Metabolic breakthrough. Natural compound targets fatty acid pathways to combat rheumatoid arthritis. A recent study published in the journal Engineering has uncovered a promising new therapeutic target for rheumatoid arthritis, RA. Researchers have identified that a natural compound, obiquilictone, OL, can mitigate the disease not by broadly suppressing the immune system, but by correcting metabolic imbalances in how the body processes fats. Shifting the treatment paradigm. Traditional treatments for rheumatoid arthritis often focus on broad immunosuppression, essentially turning down the immune system to stop it from attacking the joints. While effective, this approach can leave patients vulnerable to infections and other side effects. This new research suggests a more precise alternative: metabolic regulation. Rheumatoid arthritis is not just an immune disorder. It is deeply linked to disruptions in lipid fat pathways. By targeting the enzymes that manage these fats, scientists may be able to address the root cause of the inflammation rather than just managing the symptoms. How obiquilictone works. The study focused on obiquilictone, OL, a molecule derived from the traditional medicinal plant Phellodendron cortex. Through complex multiomics analysis, researchers discovered a specific mechanism of action. Targeting ACOT1, OL promotes the degradation of a specific protein, called acyl coenzyme, a thioesterase 1 ACOT1, restoring fatty acid balance. By breaking down ACOT1, OL helps restore the healthy balance of unsaturated fatty acids, such as arachidonic and linoleic acids, in the body, interrupting inflammation signals. This metabolic correction disrupts key signaling pathways, specifically the JAK-STAT and PI3K-AKT pathways, which are notorious for driving chronic inflammation and tissue scarring, fibrosis. Evidence from experimental models in controlled trials involving rat models of rheumatoid arthritis, the administration of obiquilictone yielded significant physiological improvements. Joint preservation. Treated subjects showed reduced joint swelling and better structural integrity of both cartilage and synovial tissue. Immune rebalancing. OL successfully shifted immune cells from a pro-inflammatory state M1 macrophages to an anti-inflammatory state M2 macrophages. It also limited the formation of Th17 cells, which are primary drivers of autoimmune attacks. Reduction of inflammatory markers. There was a measurable decrease in critical blood markers of RA, including IL-6, TNF-alpha, and C-reactive protein, CRP. Cellular control. In lab settings, OL slowed the aggressive growth of synovial fibroblasts, the cells responsible for the painful thickening of joint linings, and triggered their programmed cell death to prevent further damage. Why this matters. Rheumatoid arthritis affects approximately 1% of the global population, representing millions of people facing chronic pain and potential disability. Current therapies often fall short of complete remission or carry heavy side effect profiles. The discovery that a natural compound can bridge the gap between metabolism and immunology opens a new door for drug development. If researchers can develop synthetic versions of this mechanism, it could lead to highly targeted therapies that treat the metabolic fuel driving the inflammation, potentially offering relief with fewer systemic side effects. Conclusion. By demonstrating that obiquilictone can regulate fatty acid homeostasis via the ACOT1 protein, this research provides a blueprint for a new class of RA treatments that target metabolic dysfunction to resolve chronic inflammation.